Okay, I think we should keep the time. So, dear distinguished guests, dear colleagues, ladies and gentlemen, it is really my great pleasure to welcome you to this special symposium organized by the Nestle Nutrition Institute to answer two important questions in respect to our gut microbiome. First, how it has been developing through the evolution, particularly emphasizing the role of industrialization and urbanization. And second, how are we shaping it on a daily basis? For example, now, while you are having this lunch, we are aware there is a huge efflux of scientific evidence, and this meeting is actually proving it, on the importance of our microbiome, how it shapes from the early days, even starting with the content in meconium, how it shapes our long-term health, lifelong health, or to address it from the less optimistic side, how it paves the way towards development of different immune disorders, autoimmune, hyperimmune, IBD, heterodegenerative, and others. And therefore, it is important to address those both questions: how it behaved during the history, what was the content, and how we can change it on a daily basis. And to address both topics, there will be two key expert speakers. The first one, Professor Jens Walter, who graduated and received his doctorate in Hohenheim, Germany, but then crossed the world from New Zealand over the United States, Canada, finally being currently the professor of ecology, food and microbiome in Corts University College in Ireland, specializing in ecology of gut microbiome, while the other question, what is the effect on diversity and function on a daily basis, will be addressed by Dr. Omar Delanoy Bruno. The first lecture and the first speaker, Professor Walters, please, now the stage is yours, and then I will introduce later the second speaker, and the discussion will be at the end after both lectures will be finalized. Thank you.
Thank you very much, Sonia, for this lovely introduction. I would like to thank Nestle for giving me the opportunity to speak a second time at this amazing conference. So I am, as just been said, I'm going to talk about evolution of the microbiome, and then I will also go into ecology to try and understand, the effect of industrialization on the microbiome. And then I'm going to show you some of our data on, changing the diet and reintroducing, what we think is a lost species on, gut microbiome ecology. Actually, the stage for my presentation really has been set today with Alessio's Fasano's presentation on the microbiome. So I think you have a very good basis, and I can probably go through some of my slides a bit fast and just focus on specific things. Yeah, I do have a disclosure here. I do own a patent on Limosi-Lactobacillus reuteri PBW1, and I will talk about that strain in this presentation here. So my outline is I'm going to give you an ecological perspective on how industrialized microbiomes are altered and the environmental factors responsible. Then I will make a case for microbiome restoration. And finally, I'm going to show you some unpublished data on our microbiome restoration attempt using a non-industrialized type diet and Limosi-Lactobacillus reuteri. So this has been touched on quite a few times today, that we are host of symbionts, and that this symbiosis has evolved over very, very long times. I would say it has evolved over millions of years. And these microbial populations, specifically in our gut, they provide critical functions to host fitness. And I've listed the three what I think are the most important one here. And I think it's really important to consider that we are not just talking about tiny little contributions. We are talking to key aspects of human biologies that have changed and altered our evolution. We've heard already fascinating talks about human milk oligosaccharides, and then some aspects of these human milk oligosaccharides, that they are a substantial proportion of human milk, that they are produced at the energy expense of the mother, and then that they are not really of direct nutritional value to the infant. So really, why would this happen? And it happens because we know that breastfeeding decreases infant mortality. Before we had antibiotics by 20 to 30%, really showing, the selection pressure and really the selective, forces on these interactions of the microbiome. So human milk oligosaccharide are a really good proof for this. I'm going to show you another proof here, more related to dietary fiber fermentation. And if you look at the anatomy of herbivores, you can see over the millions of years, herbivores have evolved these large fermentation chambers in the gastrointestinal tract, allowing them to occupy nutritional niches that they would simply not be able to occupy at all. So the symbiosis with the microbiome completely altered the entire mammalian evolution. If you compare this to carnivores, which do not require microbes to digest their meat, you can see it's really just a tube. Yeah? If you look at our closest relative, the chimpanzee, you can see that we are somewhere in the middle. So we've all evolved with a diet of that is rich in dietary fiber, and you can actually see this even if you just look at this from a 10,000 mile view at the anatomy of our gastrointestinal tract. So this results, and this has been mentioned several times, this is really a co-evolved symbiosis from which we gain a tremendous benefit. But then on the other hand, the microbiome is also linked to all of these chronic, immune-mediated diseases. And you could now consider, this is a discrepancy. Why would something that has evolved to support us now cause all of these diseases? And scientists have been looking at this, and we've looked, I think, at exactly the same curves today again. And you have to see that virtually all of these pathologies that have been linked to the microbiome show these actually patterns of an increase that is clearly linked to the onset of industrialization. And since our genes haven't really changed over this time frame, and we've heard this also today, something has to have changed, and what has been put forward, and this is now more than 10 years ago by Marty Blaser and Stanley Falco, is it possible that we have a disappearing microbiome, and that we are actually losing these kind of symbiotic function, which then predisposes us to these pathologies, and then we are not really gaining the benefits anymore. Yeah. So I was very fortunate to be involved in two of the earlier studies that actually try and test this hypothesis of a disappearing microbiome. And the experimental design there was simple, from conceptual, that you compare an industrialized population, with a population that lives a more rural, non-industrialized lifestyle. Here, the first publication I show is the microbiome in Amerindian tribes in the Venezuelan rainforest, which was led by Maria Gloria Dominguez Bello. And then another publication here that was led by myself, that was published actually within a week of each other. And we were very lucky that both of these publications made it into the research highlights of the journal Nature. In the interest of time, I only have time to show you the findings of the Papua New Guinean study, but the findings were quite similar. And this is really that it is consistently that we have a higher bacterial diversity in these non-industrialized populations if we compare them to the modern world. And if you look, this is your kind of heat map. You can see that in the US controls here, we have a chunk of around 50 species that are just completely absent in the Americans, but actually quite dominant in these Papua New Guineans. Interestingly, we only detected 4 bacterial species that are actually absent in the Papua New Guineans and that we actually have. So it seems like we are losing diversity while we are gaining very, very little in return. Yeah. So, and again, there are now dozens of these studies. It's really, really fascinating research, and this is an overview by the Sonnenberg group, comparing here these non-industrialized population with these industrialized population. This is a better diversity distance, and you can see the Papua New Guineans here are actually the most industrialized of the non-industrialized. You can see these are the Yanomami, which I just showed you which are the most kind of distinct, population. But you can clearly see this gap here, as I'm saying, that it's really the industrialized lifestyle that actually alters the microbiome, and it's consistently detected. Yeah. So what are the environmental factors and what is the ecology behind this? Why is this actually happening? And we've heard a lot today about, antibiotics, which I think clearly have an effect, but it's also, other things that you might not think about, small households, hospital deliveries, not living in larger groups anymore. Then we talked a lot today about C-sections. And then I think for us, a really important factor is sanitation. And in our study in Papua New Guinea, we actually applied ecological modeling and found that there is a much, much higher degree of dispersal in these populations. Dispersal being an ecological process by which microbes are actually shared amongst individuals. So these populations are just, sharing their microbiome to a much, much larger degree. Of course, that also comes with then higher rates of infections. And, and we've heard already today about, that I don't want to, kind of romanticize this lifestyle. Yeah. But clearly what we have here is a lifestyle that provides or that generates hurdles to symbiont transmission. Yeah. And then we have clearly the diet, which is our modern Western diet is low in dietary fiber. We've heard a lot about formula feeding, high amount of processed foods, and then high amount of sugars, emulsifiers. So what I think is we have an insufficient dietary support of the gut microbiota just by not providing substrate. So first we're struggling with getting these microbes, and we saw some really fascinating talks this morning that this actually is even detectable within Western population if you look at antibiotics and C-sections. And then after not getting them early in life, we're also then feeding them very, very badly with our modern diet. And I think what that causes then is a disruption of this mutualistic relationship. And this is the old friends hypothesis. Either are we losing these old symbionts or we are not supporting them well enough, and then we're not gaining these, benefits anymore. Yeah. So I do think that all of this, makes a case for microbiome restoration. And if you actually listen to the talk this morning, if you put something like Bifidobacterium infantis or Bifidobacterium longum subspecies infantis back into a C-section kit, for me, this is already one attempt of microbiome restoration because it is a species. It's clearly co-evolved, it's clearly a symbiont, it is actually overrepresented in the non-industrialized world. So we already saw some really nice, I think, practical examples of microbiome restoration. So, but of course we could put this further, we could actually look, so industrialization has reduced diversity, it removed, microbial symbionts that we find, also these kind of lost species, we could think about them. There is clearly a drop in microbiome functions and metabolites that are important in host microbe interaction, specifically fermentation, short-chain fatty acids, heard lots of this. Then we have detrimental species in turn and metabolites, and then, and this has also been mentioned today already, that we have these pathological interactions at the epithelial interface, specifically mucus degradation, but we've also heard today from Alicio about sonulin, also meaning markers of barrier function that are actually, decreased. Yeah. And then this is another Sonnenberg, a picture which I find quite nice. Is this kind of disrupting our interrelationship with our microbiome, causing, mechanisms such as inflammation, but also metabolic changes, and then predisposing us to disease, and then causing these patterns of chronic diseases that we are seeing. So this really is the motivation to try and do something about this. Yeah. But there have been critical voices, and it's quite fascinating to follow this discussion. So this is an article from a year ago in the New York Times. So you can see this makes actually it makes it into the general important press actually. And there the argument was, rewilding your gut is probably not the way back. This article is also to some degree driven by some scientists traveling to Africa, traveling to the Hadza, and then doing very questionable experiments like a fecal microbiota transplant on themselves, by which they put the fecal sample of a Hadza, I seriously think using a turkey baster, and injecting that in a self-experiment. So this wasn't well perceived by the scientific community, and I would say justifiably so, delivering a lot of press. Yeah. But then there are also scientific arguments, against trying to reversing these industrialized changes, and I just put a few one forward here, So first, we can't really define an ancestral microbiome. We can compare these populations with the industrialized population, but we don't know how the microbiome looked, 10 or 15 or 30,000 years ago, although there are some efforts to actually sequence, old poop samples, with some degree this actually revealed some interesting insights. So clearly, we don't know exactly what the essential microbiome is, Then there's a question, is our microbiome actually mismatched? I make an argument for it, but some scientists do actually not agree with this. And the argument here would actually be, we are not living in a non-industrialized world. We live in an industrialized world, and it could be that, are we not now matched with our microbiome to this industrialized microbiome? And this is actually a statement by Dave Railman, one of the leaders in the field, in this New York Times article. And he said, look, if I live in New York City, I don't want the microbiome of the Hadza. The environment in New York is much different than rural Tanzania. And I'm pretty sure my microbiome has figured out how to manage it. So that is an argument that is put forward. I have to admit that I don't agree with this argumentation, simply just because of the time frame on how symbiosis actually evolve. If you evolve in a certain environment for millions of years, which I think the human system has, then these changes, these environmental changes that come with industrialization were simply too fast for any, evolutionary adaptations to happen. So I do argue that we are, in fact, mismatched to our microbiome. And then a very good argument is, can we even alter the industrialized microbiome given the ecological environmental context in which it exists? So we are living in a very different world. So I've already presented this yesterday, our attempt to actually go at this, but I will focus more on the ecology today. So I've already mentioned, if we have this decrease in microbiome diversity, and it's to some degree, driven by industrialized and processed foods, can we, readjust the diet in an industrialized population and put some health-promoting lost microbial taxa back? And then again, credit goes to the Sonnenberg group for these lovely graphs here. With this, we could then potentially restore the microbiome. And the focus here is very much on dietary fiber. So I'm sorry for those who were yesterday, but I still think I repeat this. So this is what we did is a very strictly nutritionally controlled diet done with nutritionists in Alberta. And again, I want to acknowledge Anissa Ahmed for doing all the hard work here, where we designed this non-industrialized type diet. It's full of minimally processed plant-based foods, more than 40 grams of fibers, and all the meals and snacks were provided to the participants, and this was the only diet they were instructed to eat. We also added Limosi Lactobacillus reuteri to this experiment, and the reason for this is if we looked into our Papua New Guinean study, then it was very, very dominant in Papua New Guinea, but it was non-detected in US Americans, and I worked with the species for a very long time. So I think I've isolated it seriously one single time in all my work from a westernized individual. So what we did is here we actually compared a placebo with the type strain of reuteri, which was actually isolated in Germany, and we have this Papua New Guinean strain called PBW1. This is the experimental design. It's an N of 30 crossover trial in terms of the diet, and then in 10 individuals, they got one strain of probiotic, 10, they got another strain of probiotic at a single dose, and then 10 got the placebo. So this is the experimental design, and what we measure, we do all the multi-omics you can think about, but then we also measure these functional characteristics that we think is linked to fermentation, such as fecal pH, short-chain fatty acids, stool characteristics, and we do metabolomics and metabolic clinical markers. So if I present here the work on the L. reuteri strains, then you can see that these two strains come into the the gut. This is done by selective plating and by strain-specific PCR. The data is identical, and you can see they're definitely washed out. So at around 10 days after you actually give these reuteri strains, they are just become non-detectable anymore. But what is interesting, the Papua New Guinean strain colonizes with around 1.5 log higher, and it also persists significantly longer. And the interesting is that the persistence is prolonged using this non-industrialized diet. So there is really an interesting interactions of this Papua New Guinean strain with the diet. It increases its ecological performance in the gut, but clearly we can't re-establish the species in a non-industrialized gut. Yeah. We also included these reuteri in our clinical kind of models, and these reuteri administration did not change any of the clinical features that we detected. So then we looked at the microbiome, and there were quite a few surprises. So if you look at the amount of features, or species essentially, and you use as a Shannon diversity and evenness, what you find is actually there's a significant reduction using the non-industrialized diet. So we actually reduce the diversity that we actually see in the gut, and this is not something that we really expected, because it's a diverse diet, that provides all of these substrates. The hypothesis was that the diversity would go up, and this is not the case. What is interesting is then if you look at, because we have a crossover trial, we can look at half of the subjects that got these non-industrialized diet first. So you can see the diversity actually decreases and the evenness decreases, but then after they revert back to their non-industrialized diet, all of this comes back. So it's not that we are losing species, but we kind of push the system probably, to some degree, but then it actually does recover. Yeah. If you look at the shifts, and this diet is an extreme diet, has tremendous changes in the microbiome. You can see this is the hierarchical clustering, which is completely separation. These are the scientific, are the statistically significant taxa, which is almost half of the taxa are actually changed. But then again, if you look at a permanova there, which kind of clusters the individual fecal sample, then you can say the individual still very much clustered by individual. And if you do this permanova, then you can see the individuals still explain around 76% of the actual variation in the data, and the diet only explains 1.4%. So meaning, as much as we see shifts here with the diet, and this is very significant, it still explains only a tiny kind of fraction of the variation. So we still maintain who we are, from a microbiome perspective. If you look at these beta diversity shifts, again, we get very strong beta diversity changes, is compared to the baseline. And then when you go back to your non-industrialized diet, all of it goes back down to what you were previously. Yeah. If we look at functions, this is done with metagenomics. The hypothesis was because, that we are functionally redundant, that the individuality wouldn't play less of a role and the diet would perhaps play more of a role. And this is in case the fact, but it is still very much individualized. You can see this here around 60%, and the diet really still only explains around 3.4% of the variation. So as an individual, it remains the major driver, even if you look at functions. Yeah. And here, again, there's less effects in terms of alpha diversity, but you can see there's still the amount of pathways, the diversity of pathways that we detect goes down, and the amount of K-cymes goes down very significantly. So the functional diversity still reduces. And again, if we look at this temporarily, reduction happens, but then when the people go back to the non-industrialized diet, everything reverts back to where it was previously. Yeah. So what I think is really important is then to look to specific functions of the microbiome. And I already showed this, I think, yesterday, that as much as the diversity of K-cymes go down, the actually total reads of the microbiota, it actually goes up. So there are more, enzymes, genes that encode for enzymes targeted in carbohydrates, and specifically plant carbohydrates. Then we get a strong reduction in fecal pH. We get an increase in short-chain fatty acids, and the amount of acetate actually increases. And then we get a significant reduction in the ratio between branched-chain fatty acids and short-chain fatty acids. And this is kind of a sign of a reduction of protein fermentation, because these branched-chain fatty acids are the result of amino acid fermentations. Yeah. And then I mentioned this, despite the effects in the microbiome being probably not what we've expected, the clinical effects are pretty dramatic. Significant improve with high significance in CRP. Plasma glucose is substantially reduced. Markers of insulin sensitivity go up. And then I've already shown this yesterday with these very, very strong effects on total cholesterol here, and very, very clean findings actually. Yeah. And then, you know, if you then look at the actually links between the microbiome and these clinical outcomes, we do machine learning random forest regressions here, and you can see there is some good, and we are really just looking at this. We've really just started, so this is brand new data, but you can see the microbiome and the K-zyme amount, you know, they are predict plasma glucose improvements. And then I've already shown this yesterday, the amount of K-zymes predicts the improvements in inflammatory markers, specifically the ratio between mucus-directed K-zyme and total enzymes. And as I mentioned today, I find this, and you know, I was kind of fascinated by the mention of you know, the barrier function in the previous talk as an important kind of mechanism by which we kind of the dialogue is, you know, disrupted between our microbiome and the host in the industrialized life. So our data really supports this, that, you know, a mucin degrading functions are then, also this decrease is actually linked to, you know, an inflammatory markers. So I hope I'm not much too long, but with this I want to conclude. So, so the lifestyle-induced microbiome disruptions you know, might predispose humans to chronic disease. We've heard it's very hard to prove this directly, but we've heard a lot of evidence today that at least the indirect, you know, findings are actually in support of this. I would argue that there is a rationale to restore key functional attributes of the gut microbiota, and I think at least some speakers today would agree with this, because they also presented what I think are, to some degree, microbiome restoration strategies. And then a non-industrialized type diet can restore functional attributes of the gut microbiota that are linked to beneficial clinical outcomes. But well, clearly we can't, this strategy is not diversifying the gut microbiota, so it's probably not generating an ancestral microbiome. But I'm hope that this focus, you know, on functional features will be beneficial in the future. Yeah? What can you do yourself? I guess you can't eat a non-industrialized diet unless I send you the recipe book. But what you can do, and I mentioned this yesterday, Anissa was leading this effort. We did write this review here on healthy eating. So I do think that, you know, there are clearly things you can actually do to improve your interactions with your microbiome by eating the same diet. I also have to admit, I think we violated most of this yesterday evening in the tapas experiment, yeah, or tapas bar that we were, which we wouldn't have to, because there were actually tapas that were very, very healthy, but then it's still hard, you know, to then say no to the amazing, you know, Spanish ham that was also provided. Yeah. With this, I would like to acknowledge Anissa Ahmed, who was very much in charge of the nutrition experiment, and then Professor Fuyong Lee, who is now in Hong Kong, and a previous postdoc of mine who did the bioinformatics. It's a long list of students and international collaborators and the funding sources. And I would also acknowledge the Papua New Guinea Institute of Medical Research, with which we did the study in rural Papua New Guinea. Thanks again for your attention.
Oops, thank you. So as I said, there will be two different topics. The second topic is how we change our microbiome diversity on a daily basis by some nutrients or constituents of our food, which will be addressed by Dr. Omar Delanoy Bruno, who graduated in Puerto Rico University. Had his PhD studies and defended the thesis at the Washington University in San Luis at the USA, and currently doing research as a specialist in gut ecology and microbiome in Nestle. So now it's your turn. Thank you.
Thank you for the kind introduction, Sonia, and thank you for the organizers and NNI for inviting me. Can you hear me now? Perfect, yeah. For inviting me to give a talk in this Congress. Very excited to be here. I want to start talking about glycans, because I think it's an important topic, and we have reviewed some of those before today and yesterday. I want to give a disclosure again. I'm an employee of Nestle, as Sonia mentioned. The outline of my talk, I'm going to give an overview of what dietary glycans are, the role of the dietary glycans on microbiome development, the importance of human milk oligosaccharides in early life nutrition. I'm going to highlight the complexity of glycan structures in foods, specifically now that we're eating lunch, with emphasis on fibers and the use of carbohydrate active enzymes. We've heard a lot of K-enzymes. I'm going to talk a little bit about what they do and how we can use this information to better understand this relationship between foods and our microbes. And lastly, some of the key learnings and opportunities. So what are dietary glycans? I think the simplest way to illustrate them is by using these symbolic figures. I'm just going to start really quickly with the simple sugars. It's just a fraction of them, but mainly fructose, glucose, galactose. When combined with each other, you can form the disaccharides such as sucrose, lactose, maltose. But it can also get more complicated as we extend this change of simple sugars into oligosaccharides. You've heard a lot of fructooligosaccharides, galactooligosaccharides, and in many instances, this 2FL, which is a component of human milk oligosaccharides. But when we get more complex, we can get polysaccharides such as those found in starch, which is illustrated here with amylopectin. And then we have another complex glycan called arabinoxylan. And I want to pay a special emphasis on this too, because our genomes can produce proteins that can break down this complex polysaccharide, but we do not have the tools to break down this other one. So our microbes are the ones that are going to be utilizing this. And this is very relevant as we talk about food. And it's because of some of the work that has been done by Erica and Justin Sonnenberg, where they describe what host-digested and microbiota-digested glycans or microbiota-accessible glycans are. Here is just simply illustrating that, and this is just very, we can use you as a test subject. When you eat your food, the majority of the nutrients that you get are going to be absorbed by the host, but there's a fraction of glycans that are going to be accessible to the microbes, and those glycans that are accessible to the glycans are going to be the ones that we cannot break down. So this is an example of what are the nutrients that the microbes are going to compete and utilize, and because of this breakdown of nutrients, they're going to produce some metabolites that are going to be helpful to us in this symbiotic relationship we have with them. And this is what we usually define as dietary fiber. Now, there's two scenarios at the moment, and we've talked about this a lot in the past couple of days, where we have a lot of this microbiota accessible carbohydrates, again, nutrients that are of benefit to the microbes. But we also have a decrease of those, again, a decrease of consumption of fiber in industrialized populations, and these have very awful repercussions to the microbiome as well. And why is this important? We know that there are specific dietary glycans that can contribute to the healthy maturation of the gut microbiome. And we have seen this a lot today and yesterday, but essentially there are some external factors that are contributing to this maturation of the gut microbiota. I'm illustrating here how the microbe develops through time. So this is from birth, lactation, and infancy and during childhood. I'm going to pay special emphasis in two components here, which is the consumption of dietary fiber and and the impact it has on during pregnancy, but also during the transition between milk and solid foods, the weaning stage during infancy. And this just to illustrate that at the moment there's a gap in the consumption of fiber. This is taken from the US data. There's a gap in the consumption of fiber. This is the requirements are in gray, and this is the average intake at the moment. So there's an opportunity here to see how we can improve the consumption of complex glycans through fibers. And the other one is through human milk oligosaccharides that we have discussed a lot. So I'm going to go briefly over it because Ivan presented it yesterday, a lot of what we know. And just to give you an overview, human milk is composed of a mix of different types of nutrients, some of which are these human milk oligosaccharides, and they're mainly composed of three main groups, which are the HMOs that are have a silyl group, neutral non-fucosylated HMOs, and neutral fucosylated HMOs. And the important thing that I want to highlight is that more than 160 glycan structures have been described, and about 20 of them are representative of more than 80% of the total HMO mass at the moment. We did a study in our group where we're trying to understand how HMOs and what is their impact on the development of the microbiome. This was a multi-center randomized controlled double-blind study of healthy term infants in 3 different locations in Europe. The idea was to test a combination of 5 different HMOs, again belonging to the 3 groups that I mentioned in the slide before. We have 2 neutral fucosylated HMOs, 2FL and LDFT. We have 2 silated HMOs and 1 neutral non-fucosylated HMO. And we have 2 arms. This is a randomized group. So we have 2 arms where we tested 2 concentrations of the 5 HMOs, one at 1.5 grams per liter, and then a second one at 2.5 grams per liter. And we have a control group where they were just fed the base formula in the group. And what we found is that in terms of diversity, and this is an important concept that I want to bring, is that in early age, high diversity doesn't mean that it's going to be something that is good to see. What we see is that in infants that are fed human milk, their diversity tends to be lower at the level of taxa. And as you can see here at baseline, children that are exclusively fed human milk, their metric for alpha diversity is lower compared to all the groups with formula fed. But what we saw is that at 6 months of age, there's a decrease in the alpha diversity compared to control formula for the 2 groups that were receiving the 5 HMO mix. They're not quite yet close to what the human milk, but it's definitely going in the direction of what the human milk glycan group is. Another thing that we notice is this impact on bifidobacteria, and the reason why we care about bifidobacteria is because they have the tools to utilize those glycans, and I'm going to spend a little bit of time in the next slide to explain how they do it. But in essence, in this particular study, we saw that the levels of bifidobacterium were higher again in baseline in this group consuming human milk. And at 6 months of age, we see that there was no significant differences between the arms that were receiving the 5 HMOs compared to the human milk. And then there were statistical significant differences compared to the control group. And we noticed that most of the differences that we're seeing is basically an increase of Bifidobacterium longum infantis, where we see that there's increases when children were consuming this formula with the 5 HMOs. We didn't see differences at different strains of Bifidobacterium, and I think that's an important message that I want to give them. So we didn't see significance at B. longum or B. bifidum. One of the ideas of why we didn't see differences is because our microbes have different strategies for the utilization of HMOs, but also different types of glycans. And this is just to illustrate 2 of those examples we have on the left, B. infantis, and then to the right we have B. bifidum. Again, B. bifidum was one of the ones that didn't change in abundance significantly between the groups. Now, B. infantis have evolved specific mechanisms to utilize the HMOs by introducing them into the cell, and then later they can process it and metabolize it. B. bifidum, what it does is that most of that metabolism of these complex glycans are outside of the cell. And I just want to illustrate that for us as biologists, it's important to understand what are the pathways that the microbes are using for breaking down complex glycans, including HMOs, because this is going to be informative when we try to understand the nutritional interventions we do in this particular population. And just to bring the gap with the gen stock is that we know that there are differences between geographical locations of the levels of different types of bifidobacterium species. So what I'm showing you here is a figure which represents the number of different types of bifidobacterium species that are highlighted in the different colors. And then in black is any other bacterial genera that is present in these populations. And what we see is that in this case in the US, there's a higher prevalence of the abundance of bifidobacteria is less than other countries, and there's more. If you pay special attention to this light blue, there's a higher increase of B. infantis in countries like Bangladesh and Gambia that we don't see mostly in other countries, either in Europe or in the US. And I want to exemplify that the complexity of glycans is important, specifically when we rethink about the transition between infant milk and solid foods. And the reason is because in the foods that we eat, there's a lot of complexity of glycans. Here I'm just illustrating how the different types of glycans are present in plants versus algae versus fungi. And just to give you an idea, even within plants, you can see different types of structures that are going to be impactful to the microbiome, because not all of them can break them down the same way. In addition to that, when we take a look at, just taking a look at different types of fruits, this is the work of Carlito Lebrilla and UC Davis. What they're trying to develop is an encyclopedia of glycans of foods, and I think this is a very important tool that we have, because at the moment, there's a black box of information of what kind of glycans are present in our foods. And Carlito's group is doing a lot of work to try to decipher what is the complexity of glycans in different types of foods. And what you can see here, I'm highlighting in different colors the different types of sugars or monosaccharides that are represented in the carbohydrates of different types of fruits. And I'm just paying close attention to, for example, pears or guava, which have an increase of xylose in their carbohydrate component that are not present in many others. And this is very important because there are going to be bacteria that are going to be better suited to digest that type of glycan than others. So when we're rethinking about what kind of foods do we recommend or give to children, it is important to understand that the microbes are not going to be the same for everyone, and there are going to be preferences in how they see them and break them down. And in addition to that, there are many sources of dietary fiber that are currently underutilized during food manufacture. They're part of the waste streams, and there's an opportunity here to be sustainable about what are the ingredients that we're using. So how do the microbes see these glycan structures? So when the microbes are interacting with foods that are being consumed, depending on the nutrient that they're interacting with, they're going to be fermenting them and producing beneficial metabolites. One of those examples is short-chain fatty acids, and we have discussed this here a lot. And I'm using here a schematic just to represent what the tools are. What are those tools that the microbes use to break them down? Again, the use of carbohydrate active enzymes or K-enzymes, which can cleave specific structures of that glycan. I'm just going to use as an example pectin because it was mentioned before today, specifically apple pectin. And the idea is that the microbes have these K-enzymes, which are represented here as scissors, that can recognize some of those structures and break them down. And this is the thing. You are possibly not going to have some of those K-enzymes, and it's a reality. We have different types of microbial communities, and we have to know that some fibers are not going to be, they're not going to be as good for you as others. And if we understand those differences, we may have an opportunity here to be more personalized nutrition for the types of foods that we give to people. This is an example of how we can use K-signs to guide that type of understanding. This was a study that I did where we essentially enrolled participants to consume 3 types of foods enriched in fibers, plant fibers. One of them was using pea fiber. The other one was use 2 fiber types, pea fiber and inulin, and then a mix of 4 different types of fiber. And the idea was to use fibers that had very different types of glycan structures to see how this impacted the microbiome of the participants that were consuming these ingredients. This is in adults. I don't want you to think we did it in infants. This was all done in adults in the US. And what we saw is that the K-sign, so here in the heat map, I'm showing you the full change of the different types of K-signs that were significantly different in the 3 different groups. And we noticed that the majority of the K-signs that were significantly different were associated to the specific structures found on those fiber types. For example, pea fiber is enriched in a glycan called arabinine. And we saw a lot of these arabinases or arabinofuranosidases having an increase in abundance for this particular group. And just to go more detail is that another opportunity we have at the moment is that we can do analysis of residual carbohydrates in fecal samples to try to understand how much of the microbiota is utilizing those ingredients when we give them. And this can give us some insights of the preferences of the microbiome for certain types of glycans. What I'm showing you here is a cross-correlation between K-signs and fecal glycosidic linkages. And what we saw is that particularly these 2 K-signs that were present in the microbiome of participants enrolled in this study were negatively correlated with these types of structures. And I'm going to go in a minute about what they are. And they were possibly correlated with this structure. In addition, we also saw an increase of that 5 arabinofuranos in the residual carbohydrates of the fecal samples, and then a significant decrease in the other structures. And just to give you, portrayed it in a more visual way. So this is pea. The fiber that you can get from pea is enriched in this arabinine structure. So this is the one that I mentioned before, which is mainly composed of this arabinose monosaccharide. And it can be in long chain, but it can also be in different types of branches. And what we saw is that the level of the sugars that are part of the branches, the ones that are enclosed in this blue circle, are being decreased, significantly decreased, and they're highly associated with the representation of the K-signs that can break those linkages down. And then we notice an increase of this particular types of glycans. We don't know if there's a specific preference for these particular branches of this component, or if there's still more experiments that we need to do. But this is giving us some insights about what are the bioactive components within a fiber ingredient that the microbes are telling us, hey, this is what we want. Why don't you give us what we need? With that, I will just try to make some conclusions. So we know that dietary glycans are an important modulator of the microbiome. We know that better understanding of the physical, chemical, and biological properties of glycans will be of importance when considering the next generation of functional foods. Expanding the diversity and variety of foods, it's important because it will allow for more an increment of the types of tools that the microbes can have to digest them. So it's bringing to the table the different types of glycans that you can find in different types of foods. And finally, we can use K-signs as an information tool to tell us what are the preferences of the microbiome for specific glycans. And with that, I thank you.
6 to 7 minutes for discussion. So please raise a hand. Yes, please introduce yourself.
Yeah, hello. Gianfranco Rompone from Biogaia, Stockholm. I have a question for Jens. I'm really fascinated by your topic on restoration. So it seems that you have a very specific, strain-specific differences between the Papua Guinea strain and another strain, which it might come from other origins. So do you think, I was puzzled by also the fact that we cannot go back to the rural or non-industrialized life is quite challenging. So do you think going back to see these co-evolutionary microbes could be a good strategy to restore in a strain-specific manner, rather than just thinking about who is there and who is not there anymore?
So, is this working?
Yeah.
It's working? Yeah. So, I mean, we are right at the beginning of all of this. I think this is, for me, it's just a proof of concept. And it's just a proof of concept, in terms. But I do see a value in looking, what are the species, that humans have evolved with over, the long periods, and then thinking, how do they interact, and then potentially giving them back. In terms of Limosilactobacillus reuteri, is the experimental design, is one single dose, and it doesn't persist in the modern world. But of course, nobody would stop us, to actually give this more repeatedly, which is not what we've tested here. But I think even if we can't, permanently colonize them, we can probably still, use probiotics and actually expose, and probably even postbiotics, if with some of these, species and compounds, to actually then expose, humans, to these, factors if they were somehow important in our evolution.
And at the end of the day, what is important is the risk markers, which are improved with this non-industrialized diet. So do you have any correlations with these more primitive populations? Are they living longer, or do you have any epidemiological?
So what we have is we have an almost complete absence of chronic diseases. So, and that was actually mentioned in the presentation today, about the evolution. So they are not living longer. So it's almost like they've treated, or we've treated, longevity and the absence of infections, with then these spike of chronic diseases. So they definitely, as in Papua New Guinea, for example, and this was striking. I visited Papua New Guinea 3 years ago, and in the 1990s they had 0% type 2 diabetes, and there was a study in 2018, and they had 20% type 2 diabetes. And imagine what happened in the last 20 years. It's not genes that changed. It's probably not even, medication. But what changed is, of course, they started to get westernized, foods, And so the effects on chronic diseases, I think, are dramatic. And I think the hypothesis at the moment is that it's at least to some degree linked to diet microbiome interactions.
Thank you very much.
More questions? Thank you.
Question for Jens. In your study, so you had 3 weeks intervention, correct? So you have an idea actually when you reach a kind of stability of this new composition of the microbiota, and have you simply reached this, or how is it?
No, we don't really know this. Yeah, we have the 3-week period, and then we see that the shifts are incredible fast, because if you look at the clustering, actually, already I think the first sample was taken 4 days after they switched the diet, and it's already completely clustering, with the all of the other fecal samples were then taken, over the 3 weeks. So it's definitely happening very fast. What I've actually forgot to mention, and I've really just saw this morning, actually, if you look at the dynamics, of the alpha diversity dropping, there seems to be some level of recovery,
later. So it seems like the microbiome is readjusting itself, but I don't think we have the time span to make very strong conclusions there.
Yeah, actually, this was my point, because
Yeah.
you have this drop of diversity, so it may take time actually to redevelop a diversity
And
corresponding.
if you look at the data, there is at least a tendency that it seems that there is already a recovery. It could also be, of course, that the subjects might not be as motivated anymore and, might skip some of the meals or something like that. But if that didn't happen, it seems like there's some, I don't know, homeostasis kicking in or something for the microbiome to recover. Yeah.
Thank you. Just to remind us, we are talking about the single nutrient, about the single bacteria, but it is actually a food matrix that comes together to the mucosa. Do you think that really a single constituent or a single bacteria can make such a changes that we will do all these preventions?
I think.
Yeah.
Is it on? There you go. I think that the food matrix does impact the way that the microbiome will perceive these ingredients. Now we're trying to understand the components of that matrix, because at the moment there's a lack of knowledge of what are the this hidden structures that the microbiome is perceiving when they're interacting in the gut, and now we're trying to have a clear picture of what are these complex structures and how they're being processed by the microbes, what are the mechanisms that the microbes are utilizing to interact with these ingredients. But at the moment, I think we're not there yet, but it's important to understand that there might be specific structures that are of more relevance to the microbiome that we know. So in a way, an approach will be the more we consume these complex structures, the better it will be for us, because then they're going to be more prevalent in the gut.
Thank you. Why are we addressing it? Because, if you take vitamin C in apple, it makes completely different changes than if you make it if you take it in a tablet. We have to finalize. So could you please, Jens and Omar, make a final statement, just a take-home message, perhaps more practical at this, not just to finish more studies needed.
Yeah. So the practical conclusion is that I think, that the probably best thing that you can do for your microbiome at the moment is to eat really a healthier diet, a diet, that is full of these whole plant foods that have the right food matrix, and that, have all these, beneficial interactions directly to the host, but also towards the microbiome. However, I'm also acknowledging that this is probably a difficult strategy and it's not going to work on a population-wide basis. And this is, I think, where the work that Omar presented becomes incredibly important, because we do need both strategies. Yeah.
Omar?
My take home.
Perhaps mine.
I think my take-home message is that when we look into the future, into the development of what we call functional foods, it's of importance that we understand how the glycans or any other nutrients is affected by processes that we inherently do to make a product, i.e. Processes of manufacture, extraction, how these changes are impacting the nutrients that we see in foods, and how can this also impact how the host perceives it, or the microbes will perceive it as well.
So by that, I would like to thank all of you to be here in such a big number. I would like to thank our expert speakers for this discussion and for the excellent talks, and also Nestle Nutrition Institute for providing us this opportunity. Thank you very much.
Thank you very much. Thanks, Omar.